This presentation is about the case of bedaquiline, providing a brief insight into the current challenges and the future of anti-TB drug development. Tuberculosis is a global health epidemic that claims 1.4 million deaths annually and is exacerbated by the rising incidence of MDR-TB and the rising prevalence of TB-HIV co-infection. Bedaquiline, also known as TMC-207, is an anti-TB drug candidate that is currently in phase 2 trials, and it offers a novel mechanism of ATP synthase inhibition. It is also unique in that it is the product of collaboration between the private Janssen Pharmaceuticals and the publicly funded TB Alliance, presenting a new mechanism known as the Private-Public Production Development Partnership, or PDP. In this case study, we'll be looking at the limitations of current treatment, R&D challenges, economic challenges, and the pipeline changes due to PDP. TB is an airborne disease that is transmitted from person to person, and about 1/3 of the world's population is estimated to have the latent stage of the infection, meaning that they have a lifelong risk of disease reactivation. MDR-TB arises from the Mycobacterium tuberculosis building resistance to two of the first-line anti-TB drugs, isoniazid and rifampicin. So what bedaquiline does is that it binds to the proton translocating C subunit, forming a wedge between the two rotating subunits. This binding stops the rapid rotation by these subunits, consequently inhibiting the activity of the ATP synthase. So looking at the trials that bedaquiline has been part of, specifically for the mirroring studies, bedaquiline was found to be more effective than 4 months of treatment of moxifloxacin. This is very key in that a prevailing concern and a contributor to the high burden of TB is the very long treatment timeline that is required. Furthermore, for pharmacokinetics, the drug was found to be well absorbed after a single dose. And for phase 2 trials, in both stage 1 and in stage 2, bedaquiline was found to have a higher sputum conversion rate than the placebo. For TB, the current treatment timeline lasts about 6 months, but we have a unique case for TB HIV co-infection in that first-line drug rifampicin is a potent inducer of the particular cytokine P450 enzymes. This then reduces the bioavailability of the co-administered antiretroviral therapy, or ART drugs, that are used for HIV. These ART drugs are metabolized through the same pathway, and this causes a decrease in efficacy for both treatments. This thus highlights the significance of the novel mechanism that bedaquiline offers. Furthermore, for MDR-TB, there's an even longer timeline of treatment. The second-line drugs that are needed to treat MDR-TB have high cost, high toxicity. The procedures are lengthy and are tedious, and because of that, they would require advanced healthcare facilities, which resource-poor nations have a lack of. And all of this contributes to abandonment of treatment by the patient. This is a critical issue in that this increases the risk of the patient having disease reactivation, and in that patient having the disease again, he's a threat to his community and could potentially then increase the disease burden. This thus further highlights the significance of bedaquiline's potential to shorten treatment. Discovering anti-TB drugs is not easy. Genome-derived screening was used before, and that was good for finding targets, but it didn't allow for testing drugability. So bedaquiline champions the new process of whole cell screening, in that was how bedaquiline was found. Whole cell screening has been found to be more successful because it allows for identification of interactions between the drug candidate and the cell membrane proteins of the Mycobacterium tuberculosis. Limitations in clinical development for anti-TB drugs include lengthy clinical trials, and this is due to the lengthy treatment that's required for TB. There's difficulty forming combination therapies. It's already difficult enough to find a drug that by itself and to test the efficacy and the safety by itself, but to do so in tandem with other drugs is difficult. There's also an inadequate global clinical trial capacity and gaps in funding to help back up the resources that are needed for both research and for development. Thus, this poses heavy potential economic burdens, and this may cause companies to not want to invest, and that there could be potential high costs due to these challenges. But the TB Alliance estimates that the internal rate of return for new anti-TB drugs ranges from 15% to 32%. Thus, TB discovery is an attractive venture. The beneficial returns for patients are key. TB patients in developing countries lose about 20 to 30% of their annual household income because they're sick and they cannot work. The social returns for patients are also important in that because the patients are now healthy, they can contribute to society, and this is especially key for developing nations. So what PDPs do is that they incentivize pharmaceutical companies to invest in the anti-TB drug development pipeline. It alleviates the risk, it alleviates the monetary burden. So having the collaboration between non-public organizations such as TB Alliance and pharmaceutical companies such as Janssen shows that the PDPs alter the risk and cost-benefit equation, and thus encourage the discovery of new TB drugs and secures efforts to ensure that the drugs will be made accessible in the markets of the regions where they're most needed. So why is changing the anti-TB drug timeline such a pressing concern? Well, a new anti-TB drug has not been approved in the past 40 years. Thus, when you look at the current pipeline, it is not sufficient enough to enable production of new treatment regimens. The finding of Bedaquiline is great. However, there just aren't enough drug candidates currently in the pipeline to put together new treatment regimens. Thus, Bedaquiline is important in that it is not only a promising drug candidate, but it also suggests and shows how improvements can be made in the anti-TB drug development pipeline.